Hi everyone, this is the genetics lectures. Okay, let's begin with some vocab. First, heredity. What I mentioned in our lectures was that I would not be asking you to define these terms, this vocabulary, in our next exam. But you'll definitely see the words as part of a question or something like that. And so you should know them for that reason, right? But they're also pretty easy. I think everyone has heard of heredity, right? Everyone has probably said things like, I inherited this from my mom, or you clearly inherited this from your father, or something like that. So the passing of traits from our parents to our kids, that's heredity. But as you're going to learn in these lectures, there's a variety of mechanisms at play that ultimately have an effect in our physical features. And so the study of how genes get passed down to another generation and how those genes interact with each other in such a way to give you your ultimate look, that's called genetics. So studying how genes interact with each other to give you your final look, that's genetics. And the person that we give large credit for starting this branch of biology is Gregor Mendel. And Mendel was an interesting um, scientist of his time. For one, he was a monk, so he was very much a religious man. But he also did some robust work with a pea plant that he was probably fortunate to have selected. Had he selected some other plant of the time, some other crop that was economically important at the time, maybe the genetics don't work out the way they did for pea plants, and maybe he's not the father of genetics after all. But he did robust work. I mean, 28,000 pea plants is a lot. But the thing that I suggest to students is to remember this, that the work that he did, the observations that he made about the characteristics or the traits of those plants, like having purple flowers versus having white flowers, being tall versus short, having a green pea pod versus a yellow pea pod, all of those traits that he was tracking in 28,000 pea plants, after a while, were showing that they had patterns in the way that they would show up in generations. He would cross this plant with that plant, and he could eventually predict what he would expect in the offspring. So, genetics is like that. You want to, at some point, find the patterns that are repeatable so that you can make predictions on your own. This is the goal. The goal is to be able to make predictions. And you can make those predictions if you understand patterns and if you can see that those patterns are repeatable. That's basically the characteristic of a pattern. It's repeatable. Okay. Um, this is the sort of work that he would do. He would take a field of pea plants, all of which had purple flowers. Just an entire field of pea plants, all of them with purple flowers. And he would have a separate field that would have all white flowers. And in those days, we would say that those plants were true breeding. In our world, with animals, it's like saying pure breed, right? If you cross a boxer with a boxer, you get more boxers. Similarly, if you cross a purple flower plant with another purple flower plant, and by crossing I mean cross-pollinating, you would find that you would create all purple flowers. And if you did the same with whites, crossed a white pea plant with a white pea plant, you would get all white plants also. So the whites field of flowers were also true breeding. We're going to learn what true breeding really meant as we learn more genetics. But this is also, again, like saying pure breed, the way we say it for animals. Okay, so when Gregor Mendel would take a purple flower and cross it with a white flower, what he would do is first he would cut off all the male parts. That's what the scissor is showing. You're cutting off all the male parts, and that way this particular purple flower wasn't able to pollinate itself. It wasn't able to breed with itself or mate with itself. He would first castrate them and make them strictly a female flower. And then he would take a brush and he would collect some pollen from the white flower and he would transfer it over to the purple one. And thus he would cross-pollinate a true breeding white with a true breeding purple. And after the pollinization, the eggs in the purple flower would all be fertilized with the pollen, and they would create all of these little pea seeds. He would then take the pea seeds, all of them are siblings of each other, and he would plant them, right? And then wait for them to grow, wait for them to produce flowers, and then he would record the color of the flowers. And as you can see, they're all purple. So Mendel decided to call that dominance. He would say purple dominates over white. Okay, so now he's developing an understanding of genetics, even though he has no idea what DNA is, so therefore he doesn't know what genes are. So he comes up with his own language, characteristics, like flower, color, characteristics, and then he comes up with variations that he calls traits, right? And he did that for a variety of characteristics. All right, so before we get into those terms, characteristics and traits, you should know right now that true breeding means these are varieties of plants that are able to self-fertilize so that they are producing identical offspring to the parent. So again, a purple crossed with itself would create more purples, right? Whites crossed with themselves would create more whites. But if he took a purple and he crossed it with a white, like we're seeing here, he would create hybrids. These would be the hybrids. And notice that the hybrids are also called F1s. These are the offspring born, or the children born, to these parents that we're keeping track of. So you're going to see a lot of parental generation, or P generation, and then you're going to see offspring generation, or what is otherwise known as the F1s. Okay, so hybrids are made from cross-fertilization, or what you could also call hybridization, right? By doing these crosses. And again, the P generation are the parents. The children of those parents are called the F1s. And if the F1s grow up to have F2s, then these are like the grandchildren to the grandparents. This is just the language of genetics. But we also have the layman language, the language that we use in society that is not too scientific. Okay, let's look at the characteristics of the pea plant. The pea plant had flower colors, and you just saw an example of the purples and the whites and crossing those two. We call those traits. They're basically alternate versions of a character, right? Later, you'll see that these are going to be called alleles. All right, what's another character? Flower position, seed color, seed shape, pod color, stem length. So there's 7 characters here. Those characters in today's modern biology are known as genes. So there's a flower gene, a flower color gene, I should say, a flower position gene, a seed color gene, seed shape, pod shape, so on and so forth. And every one of those genes comes in 2 forms. And we call those forms not traits anymore. We call them alleles. So there's a white allele. There's also a purple allele for the gene that controls flower color. There is a gene for flower position, and the 2 versions, the 2 alleles, are axial and terminal. There's seed color. It could be yellow or green. See, for every one of these, there's two alleles, two versions of the gene, two traits of the character. So traits and characters are the language of Mendel. Today, we use genes and alleles. All right, so we now know some language of genetics. So again, this is just to highlight the character and the trait, right? Let's go back to that cross where we created a hybrid. So here, again, remember, he's got a true breeding purple, a true breeding white, and when he crosses those two, he creates the hybrid. And the hybrid is also purple. And again, this informs them that purple dominates. Interestingly, when he takes an F1, this guy, and he crosses it with another one just like it, a hybrid purple crossed with a hybrid purple, he gets something interesting. Down below, you'll see the F2s. And 3/4 of those F2s are purple. And interestingly, 1/4 is white. Okay, so how's that possible? What is the genetics behind that inheritance pattern? Right, so I'm using vocabulary here to help remind you of those terms. Okay, let's go over it, but this time we're going to go over it using what we understood from meiosis as we transfer or as we transition to genetics. Okay, here's what we learned from meiosis. We know that every individual has 2 chromosomes. That includes those pea plants. Pea plants have a mom and a dad, just like you and I do. In the case of the purple one, it had a gene on that chromosome and a gene on that chromosome, and that gene was producing a purple color, and so was this one. And we crossed it, that's a little x like multiplication, with a white that also had 2 genes, one on each chromosome. This is for the white flower. But this one had a white allele, or a white color, that we learned is recessive to the dominant purple. So we're going to use little p, I'm going to put a tail there, to indicate that they are recessive. So indicating that they're small or lowercase p's. All right, here's what meiosis has taught us. It taught us that the loss segregation separates those chromosomes and that only one of those will end up in the child. And in reality, it's a sister chromatid, right? Not the entire chromosome. But remember that the chromosome is made up of identical sister chromatids. So we can just leave it like this for now. We know that this parent is going to contribute an X with a big P, and we know that this parent is going to contribute an X with a little p. And because purple dominates, all of these F1s are purple. Okay, but then this F1 generation grows up. Remember, these are the parentals, or the parent generation. And they've just been crossed to create the hybrid. Oops. Here. So they're going to grow up, and they're also going to obey the loss segregation when they do meiosis to create their own gametes. So this parent, right, will do the loss segregation and give either the big P or the little p. So let's imagine that we have 2 hybrids, one with a big P, one with a little p. Give a little tail. And we cross it with another hybrid also with a big P and with a little p. And we do the loss segregation for each. Okay, how can we demonstrate that you can get a big P or a little p from one parent? You can get a big P or a little p from the other parent. This is where we use the Punnett square. It kind of helps organize things. And also, Let's just forget the chromosomes. Notice that the chromosomes all just look like X's the entire time. There's really no value to them. The more important thing are the genes, the letters that represent something. Remember that big P represents purple, right? Remember that little p represents white. So let's forget the X's. What would this parent contribute in the Punnett square? If the loss segregation takes place, they can give a big P or they can give a little p. What about this parent? Loss segregation takes over, they can give the big P or they can give the little p, right? Okay, what is it that these letters represent now? Well, for one parent, the letters represent the genes that are found maybe on an egg, and for the other parent, they're the genes that are found maybe on the sperm, right? It's pollen, but I'm just going to draw a tail to make it obvious that it's like sperm. If this big P is given to this egg with this big P, then you will have a child that has big P, little p. That big P drops down, that big P moves over. If I go to the right of that to fill in this box, the big P goes there, the little p drops down. If I go down to this box over here now, the big P drops down from the top, and then the little p moves over. I hope you're following along. So at the last one, it's little p, little p. Okay, so let's take a look at what the outcome is. What's in there in those 4 boxes? All right, if you start here and you work your way all the way around, right? Maybe I can use red. Start here, and then go 2, and then go 3, and then go 4. You can report what you see, okay? First, we would notice that in the first box, there's a big P, big P. And that came from that guy and that guy. In the next box over, we see that there's a big P with a little p. Big P, little p. The little p came from right there. The big P came, again, from right there. They just move over. And notice that there's 2 of them. So there's 2 of those and 1 of those. Just 1 of those. And then finally, there's a little p, little p. I'm going to do this one this way. And there's only 1 of those. And if you were to count in that box of 4, how many are purple and how many are white, you would notice that there are 3 that are purple and there's only 1 white, right? So let's go back. See down below? 3 out of the 4 in the box are purple and 1 out of the 4 in the box is white. All right, so I mean you can rewind to get that entire sketch. Let's do it one more time. But this time, let's leave the chromosomes out. Let's try and clean it up. So you had a purple flower. Oops, actually there it is. Let's go this way. Oops. A purple flower. That was true breeding. Big P, big P. And you had a white flower. That was little p, little p. And when you cross them, let's just do a Punnett square. You can put the big P there and the big P there. So one goes there, the other one goes here. And then you had little p, little p. One little p goes there, the other one goes there. And then all you have to do is just kind of using a graphing style, bring them together. That big P goes there, that little p drops down. Right? Oh, I'm sorry, this is the big P. No, this is the little p. And then big P with little p, big P with little p, big P with little p. They're all the same. So what am I showing you here? I'm going to go back one slide. I'm showing you this cross. Big P cross with little p. And what the outcome is that all of them are big P with a little p. And because they have that big P, they're all purple. Because purple dominates over white. That's what Mendel saw, that's what he learned. And then when he took a purple and he crossed it with a purple of the hybrids, right? These right here. It was like getting big P, little p crossed with big P, little p. And if you do that Punnett squared, let's do that one bigger. The big P there, the little p there, where'd they come from? Well, the little p is that one, and the big P goes there. Big P goes here, little p goes there. And then they come together. Big P, big P, big P, little p, big P, little p, and then over here, little p and little p. And again, if we go in here and we take inventory, big P, there's one of those. Big P, there's two of those. There's the two. And then little p, there's one of those. How many of them have at least one big P? Well, that one has at least one big P. That one has at least one big P. And there's one of those and there's two of those, so that's a total of 3. So 3/4 are going to be purple. And then 1/4 is white. All right, those are the crosses, or those are the F1 and the F2s, right? This would have been the F1, these are the parental generation, and then these are the F2s. Okay, you might want to go back and review that. Let's keep going. This is what we saw. We saw big P, cross with little p. This parent can only give a big P, this one can only give a little p. So they're going to create offspring that are big P, little p. But this flower can give you a big P or can give you a little p, right? It has both. And if you cross it with another one just like it, then they can also give both. So I think that the diagram down below, the Punnett square down below, this one, really shows it well, right? Purples and the whites, 3/4 purple, 1/4 white. But there's some vocab to add right there and right there. What's a genotype and what's a phenotype? Okay, let's write some genotypes down as we see them here. All right, so here's one genotype. It's big P. That's literally a type of genes or a combination of genes. So that's a genotype, like genes carried, or literally type of genes carried. And there's also a big P, and there's also little p. Every one of those is a genotype. There are 3 genotypes listed there. And what does this genotype lead to in terms of a color? It leads to purple, because there's at least one dominant allele making you purple. And here, that's also purple, because there's at least one dominant allele making you purple. So that also becomes purple. And this one, on the other hand, doesn't have a dominant capital P. It only has little p's. So this one turns out to be white. Your physical appearance, that's your phenotype. So you should know from here on out that your genotype leads directly to your phenotype. The type of genes you carry determine what your physical look, your physical outcome will be. Genotype leads to phenotype. Okay, study that, and then I'll do another recording, and you can move on to that one when you have your vocabulary down and you understand this P generation giving you the F1 generation, giving you the F2 generation.